Dear students, now I am going to explain you plasma membrane structure, fluid mosaic model. You have seen the ultrastructure of cell in your previous classes. You know it is the entire cell in the scanning electron microscope micrograph. What you see here, this is the intestinal cell, and this you will find it may be any other cell, it may be a plant cell. It is the main structure. What you see, it is the membrane, plasma membrane, and inner membranes of endoplasmic reticulum, Golgi complex, then nuclear membrane, the membranes in the mitochondria. It is a membranous compartmentalization what you see in the cell inside the eukaryotic cell. So, how about the structure of the plasma membrane? So, plasma membrane or cell membranes are lipoprotein, lipids and protein, living dynamic structure. They are not a static non-living structure or a semi-permeable membrane. It is a living dynamic membrane, or it is a selectively permeable membrane. It selects the molecule which has to be transported. So, that is what selectively permeable barrier existed around the cell, and as well as the integral part of all cell organelles what you have seen in the picture. Plasma membrane is the outermost envelope surrounds the cell which separates and protects the cell from the external environment, and provides a connecting system between the cell and its environment is called plasma membrane. So, it connects with the other cell also. The fundamental structure of the membrane is the phospholipid bilayer. It is the main the constituent of the membrane is the phospholipid bilayer, which forms a stable barrier between the two aqueous compartments. So, outside and inside aqueous compartment, it is separated, or it is the plasma membrane, the phospholipid bilayer acts as a barrier. In the case of plasma membrane, these compartments are the inside and outside of the cell, and in other cell organelles, it compartmentalizes the cytoplasm, because cytoplasm is a aqueous medium. It is compartmentalized with the help of these the membranous structure, endoplasmic reticulum, Golgi complex, mitochondria, then nuclear membrane. All these compartmentalize this the cytoplasm. Proteins embedded with this phospholipid layer carry out the specific functions of the plasma membrane, including selective transport. So, it is a barrier. So, how the molecules can be transported across the membrane, it is there the proteins. So, these proteins are carry out this transport, selective transport across the membrane, and also they play a main role in cell to cell recognition. You know that and adhesion also, the cell to cell adhesion, recognition, the antigen you have studied already immunology. So, the plasma membrane is composed of two layers of phospholipids back to back. You will be able to see this is the one layer of phospholipid and this is two layer of phospholipids back to back. You can see this phospholipid molecule in this form. This is the polar region, and you can see you will find the polar head you will find, and these are the non-polar fatty acid tail. So, that is how they are arranged in the back to back. Phospholipids are lipids with the phosphate head. This is the phosphate head, and attached to them. So, these the principal components of plasma membranes are the lipids, phospholipids and cholesterol. So, these are the major lipids constituent of the plasma membrane, and also you will be able to see here. So, these are the phospholipids, they are forming the main backbone of the membrane. Then in between you will find the proteins are inserted in this, and also you will find the carbohydrates. You will be able to see the These are the carbohydrates. So it's a two layer lipids. Each lipid contains a hydrophobic or water repelling tail. This tail portion, whatever you will find, the middle part. So this one is a hydrophobic in nature, whereas the outside, the polar head, this is hydrophilic in nature. So this that's why in the bilayer, what you see, the one side it is that's why it is called amphipathic molecule. One side it is attached to the water molecules, or else it is hydrophilic, and the other side of the same molecule is hydrophobic. So the hydrophilic heads of the the hydrophilic region on the either side of the bilayer interact with the water rich environment on either side of the bilayer. Phospholipids have a polar head, what I see you will find here. So this is how the phospholipids, they have a polar head, and two hydrophobic hydrocarbon tail, you can see hydrocarbon tail, fatty acid non-polar tail, hydrophobic in nature. The tails are fatty acids and they can differ in length. So it is the fatty acids, whatever you will find in this tail, they contain 14 to 24 carbon atoms. They may be saturated, or there may be fatty acid saturated or unsaturated. Excuse me. Phosphoglycerolipids consisting polar hydrophilic phosphate, you will be able to see this is the phosphate group, phosphate head, and there is a glycerol neck, this is the glycerol neck, and non-polar the hydrophobic fatty acid tail. So this is the basic structure of the membrane phospholipids. There are about the 4 types you will find. They are phosphatidyl choline or lecithin, phosphatidyl ethylamine, cephalin, phosphatidyl serine, and phosphatidyl inositol. So usually you will find in the plasma membrane phosphatidyl choline. So it is in the neurons you will find phosphatidyl ethylamine or cephalin. So it varies, you can see these are the different types of phospholipids. All they have the basic structure, they have a polar head phosphate group and glycerol neck, and the unsaturated or saturated fatty acid tail, which is non-polar hydrophobic in nature. Head region is polar hydrophilic in nature. So this is what you can see clearly. This is intracellular and extracellular. So the polar head and hydrophobic region. So membrane proteins, there are we discussed so far phospholipids. Another important component of the membrane is the membrane protein. You will be able to see they are inserted. So they may be, you can see the there are 2 groups. So extrinsic, they are found in the peripheral region. You will find here. So they may be peripheral, extrinsic. They are loosely associated with the polar heads of the phospholipids at inner and outer surface, involved in cell signaling. Peripheral membrane proteins are often associated with ion channels and transmembrane receptors. And there is also another class of protein, intrinsic or integral membrane protein. You will find these are integral proteins. So these integral proteins are transmembrane proteins. They span the membrane and they connect outside environment with the inside. This is transmembrane, you will find. They may be transmembrane, extending through the lipid bilayer, contacts the interior of the cell and touches the exterior also. You will find they are they are found in the exterior, interior, and also they span the lipid bilayer. So that is what you can see. Then integral proteins attached to the membrane only one side you will find. And proteins are transporting molecules, acts as a cell receptors. They have a many functions we will discuss later. So they may be spanning 4-5 times, you can see like this here, only one time spanning, you can see different types of integral or intrinsic protein. So, these membrane proteins depending on their functions, they are called permeases. They are carrier proteins or integral proteins which bring the transportation across the membrane. So, they are transporter or else they are carrier molecules. They may be receptor protein, integral protein which functions as a cell surface receptors, recognize the chemical messenger and bind to them. Mainly the hormone, you can see they acts as a receptors. They bring intracellular signal transduction. Then they may be antigenic. So, they have a specific membrane proteins, example antigen A you will find on the RBC membrane. They may be antigenic, they may be enzyme, they make ATPase, catalyze the chemical reaction, they may be enzymatic in action, or they may be adhesive in action, or they may be anchoring. So, you will find different class of proteins. So, this plasma membrane, the structure, it is now the well designed structure, that is what fluid mosaic model which is proposed in 1972. Singer and G. Nicholson proposed fluid mosaic model to explain the structure of biological membranes. This fluid mosaic model is the most accepted model now. So, according to this, membrane is viscous fluid. It is a fluid, it is a viscous fluid because it is a phospholipids, it is a oily or viscous fluid, phospholipid bilayer, in which globular proteins are inserted in a mosaic pattern. There is no definite pattern, you can see it is a mosaic pattern, and also they keep on changing their position also in this membrane itself. The distribution of lipids, proteins and carbohydrate give a mosaic appearance to the structure. So, you will be able to see this is the fluid mosaic model, and you will see the phospholipid bilayer, and these phospholipids are restricted their movement by the presence of the cholesterol, you can see. Then these are the integral proteins, they span 2 or 3 times, you can see. They may be carrier proteins, or there may be protein the channel proteins, they are ion channels. So, they may be antigenic proteins, glycoprotein. So, different types of proteins, and in the cytosol you can see they are anchored to the cytoskeleton. So, this is what you can see the fluid mosaic model. So, fluid mosaic model of Singer and Nicholson is now widely accepted model which proposes that integral membrane proteins are embedded in the phospholipid bilayer. Some of these proteins extend all the way through the bilayer and some are partially across it. These membranes proteins acts as a transporter proteins and receptor proteins. So, this model you can see their model proposed that the membrane behaves like a fluid rather than a solid. So, the proteins and lipids of the membrane move around the membrane much like a bios in the water. It is such movement causes a constant change in the mosaic pattern of the plasma membrane. You will be able to see this the movement of the fluid mosaic model, where you can see how the molecules get into the water molecules get into the membrane, and the movement of the protein, can you see they change their position. They are not static, they are keep on changing their position. So, this is compared with the something like the fluid mosaic model explains the plasma membrane is a flexible membrane of phospholipids where proteins are embedded like a floating iceberg. You will find this is a water and this is a floating iceberg. So, this depending on the water waves it is keep on moving in the sea. So, similar the comparison with respect to the plasma membrane or else any other biological membrane. So, this is what you can see the mosaic pattern. So, you can see the proteins and carbohydrates, even the lipids, they keep on moving, that is why their organization keep on changing, that is why it is called mosaic pattern. Membrane is viscous fluid phospholipid bilayer in which globular proteins inserted in a mosaic pattern. In this phospholipid bilayer, hydrophobic tails of the fatty acids form the middle core. You will be able to see this is the fatty acid, they form the middle core. Hydrophilic heads line both the surfaces, you can see that. Phospholipid molecules are highly mobile, you have seen that in a previous slide. Showing rotation, lateral diffusion, flexion, flip-flop movements, both the phospholipids. So, the lateral movements are the one, they are moving laterally like this. So, flip-flop means they move from here, they change their position. Then flexion means their fatty acid tail, they get this is the fatty acid tail, it get reduced by flexion. So, all these movements are you will find in the phospholipids. Both phospholipids and integral proteins are amphipathic in nature. Amphipathic means they have water hydrophilic outside, all these molecules, and hydrophobic in the middle, not allowing the water to get inside freely. So, that is what you can see. So, this membrane proteins show lateral diffusion and interact with lipid molecules. So, it is the protein also make lateral diffusion. You will be able to see this is the carbohydrate and these are the cholesterol and phospholipids. So, oligosaccharides either bind to the protein, or you will find, or to the lipids you will find here. They are glycoproteins or glycolipids. Carbohydrates are hydrophilic, hence they are present outside, that is why they are not inserted inside the middle of the membrane, they are found in the outside, you will find. Carbohydrates restrict the movement of the lipids and the proteins. So, the carbohydrate and cholesterol, they restrict the movements of the lipids and the proteins. They play a key role in cell recognition because they are associated with the glycoprotein, they are functioning as antigen, cell recognition, antigens, and major histocompatibility molecules, they play major role in immune cells. Degree of saturation, unsaturation of fatty acids, the presence of the cholesterol, you will find these are the cholesterol. Carbohydrates regulates the fluidity and movement of the molecules, they offer rigidity to the structure. So, then hydrophilic heads forming the inner and outer surface of the molecules, you know that. So, which keeps constant water circulation. So, either side, the outside and inside, the water circulation will be there because of they are hydrophilic, whereas the middle portion is hydrophobic. Fatty acid core acts as a selective permeable barrier, save the cell and organelles from osmotic shock. So, the water molecules cannot enter freely here, it can enter through aquaporins. These are a kind of membrane proteins, function as a water channels that play a critical role in controlling the movement of the water across the cell. These channels are widely distributed in all kingdoms of life, including bacteria, plants and the mammals. So, dear students, membrane is a very beautiful collage of proteins and other molecules embedded in the fluid matrix of phospholipid bilayer. So, this is the model given by Singer and Nicholson, this fluid mosaic model. You will see the presence of you will see the phospholipids and the fatty acid tail, and these black ones are the cholesterol. Then extrinsic proteins, intrinsic proteins, then the carbohydrate moieties attached to the proteins or lipids, all these you will see. It is a beautiful collage of proteins and phospholipids. Thank you.